Hello, everyone. I'm Roland Walter, and I work at the Hutchinson Cancer Center in Seattle as a physician scientist focusing on translational clinical research in AML. One of my interests is centered around measurable residual disease, or MRD testing, by multiparameter flow cytometry. It is now well established that MRD, as detected by multiparameter flow cytometry, identifies a subset of patients with worse outcomes than patients in the same treatment situation in whom no MRD is found. The association has been shown across the entire AML treatment journey. The outcome difference between MRD positive and MRD negative patients is quite substantial, leading to increasing interest in using MRD data to guide decisions regarding subsequent treatment. Multiparameter flow cytometry works in about 90% or so of AML cases for MRD testing. However, the assays that are currently used differ widely between laboratories regarding many characteristics. Therefore, it is likely that the performance characteristics of individual tests vary. Yet, based on guidance from the FDA, the European LeukemiaNet MRD Working Group currently defines MRD test positivity uniformly at the 0.1% level. This begs the question whether this assay-agnostic uniform definition of MRD positivity leads to a suboptimal discrimination of relapse risks. We recently examined what the prognostic significance of individual MRD tests is when different positivity thresholds are used. We also wanted to empirically determine optimal MRD positivity thresholds of individual assays, and we wanted to understand what the impact of different positivity thresholds is on how MRD assays perform. In a collaborative effort with investigators from GIMEMA, HOVON, and the MRC NCRI, we put together 4 data sets comprising a total of almost 2,400 patients. 3 of the data sets contain data from patients undergoing induction post-remission chemotherapy, whereas 1 data set contained data from patients undergoing allogeneic hematopoietic cell transplantation in either first or second morphologic remission. We drew 3 main conclusions from our analysis. First, patients with MRD less than 0.1% had higher relapse risks than MRD undetectable patients in 3 of the 4 cohorts. In fact, the outcomes of these patients were quite comparable to those of patients with MRD of 0.1% or more. These relationships remain similar after multivariable adjustment. Second, we found a nonlinear relationship between MRD burden and relapse risk, with relapse risks reaching a plateau at generally low but variable levels of MRD, and increasing levels of MRD not being associated with proportionally high relapse risks. And third, optimal MRD thresholds differed from cohort to cohort, ranging from 0% to 0.08%, so very close to this 0.1% arbitrary threshold. Using these optimal thresholds minimizes the number of high-risk patients misclassified as low risk in the MRD negative group. Together, these data show the economization of MRD results at any arbitrary threshold may optimally stratify relapse risks. A one-size-fits-all approach is not ideally suited in discriminating relapse risks of patients in the best possible way. Indirectly, our findings support centralized MRD testing as it allows, unlike testing across many low-volume centers, establishment of optimal assay and context-specific MRD positivity thresholds that can then be applied in prospective trials.